పెన్ ఇక్కడ వచ్చాను. పెన్ అది స్క్రోల్. మీరు స్క్రోల్ కావాలంటున్నారా?
స్కూల్ కావాలి.
దాని అక్కడ కిందికి ఇట్లా
ఇట్లే రావట్లేదు.
కిందికి వస్తుంది. అనేది దాంతో ఎట్లా దాంతో పడతాను.
రైట్.
బ్లాక్ రైట్. ఇది చేసిన అక్కడ ఆపోజిట్ గా చేయాలి.
ఓకే ఓకే. Inheritance of two gene, dihybrid cross. So already we have completed the monohybrid cross. Monohybrid cross means cross made between two individuals different single character. Cross made between two individuals different single character is called monohybrid cross. But here the cross made between two individuals different two characters is called dihybrid cross. Cross made between two individuals differing two characters is called dihybrid cross. Cross made between two individuals different two character is called dihybrid cross. So dihybrid cross nothing but in between two characters. Two characters we take, one is the round. So here is the దీని మీద రైట్ అనుకుందాం.
పైన సార్ పెన్ అది స్కెచ్ పైన పెన్ అది.
ఓకే.
ఇప్పుడు బ్లాక్ సెట్ చేసుకోవాలి. మీరు అక్కడ క్లిక్ చేయండి. బ్లాక్ వైట్ పైన బ్లాక్, గ్రీన్ పైన వైట్.
ఓకే రైట్.
అది అరేంజ్ చేయండి.
అదే మళ్ళీ పోవాలి.
ఓకే ఇప్పుడు పైన.
సో రైట్. Round yellow and wrinkled green. These are the two parents. Round yellow and wrinkled green.
Fine. White. Plus.
White.
Okay.
RRYY. Small RR, small YY. These are the two parents. Female parent as well, male parent. So each parent consists of two different characters. One is round, another one is yellow as well, wrinkled, another one green. So round yellow, one of the parent, and wrinkled green other parent. These are the two contrasting traits. Round yellow and wrinkled green. Whenever we cross these are the pure homozygous plants. Always, according to Mendel, the parents are always pure, nothing but homozygous. Homozygous round yellow as well homozygous wrinkled green. Round yellow and wrinkled green. If we have hybrids, not pure, what we have to do? So continuous self-pollination result in the homozygosity. Continuous self-pollination result in the homozygosity. To get the homozygous condition, continuous self-pollination result in the homozygous condition. Then we take them as parents. Right? Homozygous round yellow and homozygous wrinkled green are the two parents. In between, cross is made. Round yellow and wrinkled green, right? Round yellow as well wrinkled green are the two parents. Automatically they produce the gametes. They produce the gamete, right? RY as well, small r, small. small y. So these are the gametes. These are the parental generation. So these are the gametes. Gametes capital R, capital Y, and small r, small y. In between these two, cross is made. Automatically we get the F1 generation. F1 generation is always hybrid, but here dihybrid. Capital R, capital Y, small y. It is the dihybrid. 100% plants show one of the parental character according to law of dominance. According to law of dominance, F1 generation always shows one of the parental character. 100% one of the parental character shows. Right. Then the F1 is self-feed. According to Mendel, the F1 is self-feed. Automatically, capital R, capital Y, small y. F1 is self-feed with themselves. Automatically, we get the F2 generation. This is the F2 generation. This is totally the F2 generation, right? So this is the F2 generation.
Fine.
Okay, right. So F2 generation, we get 16 plants. We get the 16 plants. How do we get the 16 plants? Initially, we know monohybrid cross. According to monohybrid cross, 3:1 is the phenotypic ratio. 3:1 is the phenotypic ratio, and 1:2:1 is the genotypic ratio according to monohybrid cross. So like that. మీకు ఈ పేజ్ మారాలంటే
బోర్డు
బోర్డు కావాలి.
దీన్ని క్లిక్ చేసి మళ్ళీ ఇది కావాలంటే ఇది
దీన్ని క్లిక్ చేసి అది ఇస్తుంది.
క్లిక్ చేసి ఇది కనబడుతుంది. ఇది కావాలంటే దీన్ని క్లిక్ చేసి ఇది కావాలి.
So right, RRYY, RRYY. So nothing but round, yellow, and sprinkled green. So these are the two parents. Parents are always pure. Female parent as well. Male parent. This is the parental generation. Parents are always pure. As well, they give the gametes capital R, capital Y. Always homozygous condition produce one kind of gametes. So these are the homozygous, both the homozygous. So automatically they give one kind of gametes, that is capital R and capital Y. This produce the small r and small y. Only one kind of gametes are generated because of they are homozygous. Parents are always homozygous. If they are heterozygous, automatically we can get the homozygous by continuous self-pollination. Right. These are the gametes. Right. Capital R, capital Y is crossed with small r, small y. Automatically we get capital R, small y. Capital R, small r, capital Y, small y. 100% plants produce heterozygous condition that shows one of the parental character. So it is self-feed. It is the F1 generation. F1 is self-feed. Capital R, capital Y, small y. Automatically we get here dihybrid is there. Dihybrid. One hybrid, another hybrid. So dihybrid. Dihybrid produce 4 kinds of gametes. Dihybrid produce 4 kinds of gametes. For example, in monohybrid, we take capital R, small r is crossed. Capital R is the gamete and small r is the gamete. So capital R, small r is the F1 generation. So it is self-feed. Capital R, small r. Automatically it produce capital R, small r, capital R, small r, right, capital R, small r, capital R, capital R, capital R, small r, capital R, small r, small r, small r. So this is the monohybrid cross, one of the monohybrid cross. It is one character, that is the shape of the seed. Shape of the seed. Here it is the round, it is the wrinkled, round and wrinkled. Then we get F1 generation. It is monohybrid. One hybrid is there, but here two hybrids are there because we take two characters. The plants are differed with two characters. One is the shape, another one is the color of the seed as well, shape of the seed. So we have taken the two characters. Automatically we get the dihybrid. It is the monohybrid. Here how many gametes are there? Capital R and small r. These are the two gametes are erased. But here four type of gametes are erased. Capital R is fused with capital Y, capital R with small y, small r with capital Y, small r with small y. This is the combination of alleles. Capital R may go with capital Y, capital R may go with small y, small r may go with capital Y, small r may go with small y. It is the independent assortment. What is done here? Nothing but independent assortment. So I will explain later. Right. So right, automatically it produce same type of gametes, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. So these are the four type of gametes are there because of dihybrid. Dihybrid always generates four type of gametes. Homozygous always produce single type of gamete. So it is the double homozygous. Automatically that produce one type of gamete. These are the homozygous automatically produce one type of gamete. This is also homozygous produce one type of gametes. But here double homozygous produce also one type of gametes. Always homozygous condition. The parent is homozygous. They produce the gametes as one type of gamete. Right, here also it is double homozygous. Automatically they produce the one kind of gametes. Small r, small y. Nothing but wrinkled and game character. And each allele and each gamete will carry only haploid set of chromosomes. Nothing but one genome. Nothing but one allele of a chromosome. Each gametes is always pure. According to Mendel, law of purity of gametes is there. Automatically gametes are always pure because they carry only haploid set of chromosomes, nothing but one allele of one chromosome. Automatically gametes are always pure. Right, each gamete carry only one allele of round, one allele of yellow. Automatically one allele of wrinkled as well, one allele of green. So gametes are always pure. But here dihybrid automatically produce four type of gametes. Here gametes are also pure. One gamete carry only capital R and Y, and round and yellow. Automatically another gamete carry 50% of the choice is there. Every character, every trait has the 50% of the choice to enter into the gamete. Right. So right. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. These are the gametes. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. When we placed in a Punnett square, so right, when we place it in a Punnett square, automatically we get the data. Right here, 16 boxes are there. So the phenotypic ratio is so right. So right, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y, right, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. These are the gametes, and the left hand above the square. Automatically we get capital R, capital Y, capital Y, capital R, small y, capital R, small y, capital R, small r, capital Y, capital R, small y, capital R, small r, capital Y, capital R, small r, capital Y, small 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 y, right, capital R, small r, small y, small r, capital Y, small r, capital Y, small y, small r, capital Y, small y, small r, small y, small y. So totally 16 boxes are there, in which the phenotypic ratio is 9:3:3:1. Genotypic ratio is equal to 1:2:1:4:2:1:1:4:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1:1::::::::::::::::::::::::::ّ. Right. So 9:3:3:1 is the phenotypic ratio, as well 1:2:1, 2:4:2, 1:2:1 is the genotypic ratio. How do we get these ratios? According to monohybrid cross, in monohybrid cross, for example, capital R is crossed with small r, automatically we get capital R as well small r. When we cross, the F1 generation is capital R, small r. It is self-fed, capital R, small r. Automatically we get the capital R, small r, capital R, small r of the gametes. F2 is the capital R, small r, small r, automatically small r. So these are the three plants are round. It is the dominant character. Three plants are round, as well one plant is wrinkled. So it is the 3:1 generation of phenotypic ratio. Phenotypic ratio is 3:1, in which 3 are round, 1 is wrinkled, wrinkled, right? It is the parental generation. These are the gametes. It is the F1. These are the gametes. Automatically we get the F2. Yeah, as well, same. Capital Y, it is the another character is crossed with small y, small y. Capital Y means yellow color. It is the green color of seed. Yellow seed, green seed. It is the round seed, wrinkled seed. Round seed as well, wrinkled seed. So it is one of the monohybrid as well. It is one of the monohybrid. Automatically we get the capital Y is the gamete as well. Small y is the gamete. It is the parental generation. It is the gametes. Automatically we cross. Automatically we get the capital Y, small y. It is the F1 ratio. F1 is always hybrid. That is 100% automatically. When it's self-feed, capital Y as well small y, automatically we get the gametes. Capital Y, small y, capital Y as well small y. So the combination is capital Y, capital Y, capital Y, small y, capital Y, small y, small y, small y. So here also the phenotypic ratio is 3:1. So 3 is the yellow color and 1 is the green color. 3:1 ratio, 3 yellow color and 1 green color. Same phenotypic ratio appeared here. It is one of the monohybrid cross. It is one of the monohybrid cross. But here we take two crosses at the same time. Two characters cross made between the individuals differ in two characters. So automatically we combined these two characters in the cross, in the dihybrid cross. Right. 3:1 is the phenotypic ratio and genotypic ratio is 1:2:1. So 1 is the homozygous yellow and 2 is the heterozygous yellow, 1 is the homozygous green. Here also genotypic ratio is 1:2:1. So 1 is the homozygous round, 2 are the heterozygous round, 1 is the homozygous wrinkled. 1:2:1 is the genotypic ratio as well. Same genotypic ratio 1:1 is there. It is the color of the seed. It is the shape of the seed. If we go to dihybrid cross, dihybrid 9:3:1 is the phenotypic ratio. Phenotypic ratio. So 3:1 is the one of the character of phenotypic ratio, and another character of the phenotypic ratio is 3:1. Automatically we get the 3:3, 9, 3:1, 3, 1:3, 3, 1:1. So it is the dihybrid phenotypic ratio. So these two are monohybrid phenotypic ratio. Monohybrid into monohybrid result in the dihybrid. One monohybrid phenotypic ratio is into the another monohybrid ratio. Because of two characters we take in dihybrid. That is why one of the character phenotypic ratio into another character phenotypic ratio combined to form dihybrid ratio. That is the 9:3:3:1. Here 9 plants are round yellow. 3 plants are round green. 3 plants are wrinkled yellow. 1 plant is wrinkled green. Wrinkled green. 9:3:3:1. Totally How many round seeded plants are there? How many round seeded plants are there? How many round seeded plants are there in F2? So totally round seeded plants. So it is the round 3 by 4, 1 by 4, 1 by 4. So 3 plants out of 4. 3 plants are round out of 4. 1 plant is wrinkled. 1 plant is wrinkled. Right. 3 by 4. Here totally 16 plants are there. Total plants are 16 in dihybrid cross into 1 by 4, 3 by 4, right. 4 1 jar, 4 jar, 4 into 3, 12. So total 12 plants. 12 plants are round seeded. So it is very, very important. 12 plants are round seeded. So 9 are round. Automatically 3 are round. 9 and 3 different combination. 9 plants are round seeded as well. 3 plants are round seeded. Right, round seeded plants are totally 12 plants. As well, wrinkled seeded plants are 1 by 4, 16 plants. So 1 by 4, 4 jar 16, right. 4 into 1, 4 plants. 4 plants are wrinkled. Wrinkled like this. How many yellow plants are there? Same ratio. Yellow plants are, according to monohybrid cross, 3 plants are yellow, 1 plant is green. Automatically, yellow is 3 by 4 out of 16, right? 4 jar, 4 3 jar, 12 plants are there. Same. Yellow seeded plants are there, 12 as well. Green plants are there, 1 by 4 into 16, 4 jar, 4 1 jar, 4. 12 plants are yellow, 4 plants are green as well. 12 plants are round, 4 plants are wrinkled. So this ratio occurs because it is dihybrid cross. Dihybrid cross is nothing but 2 monohybrid crosses unitedly result in the dihybrid cross. Here we take the 2 characters. One is the seed shape, another one is the seed color. Seed shape as well seed color. Automatically we get the ratio. Totally 16 plants. In monohybrid cross, we get only F2 4 plants are there. But in dihybrid cross, we get the 16 plants. Nothing but monohybrid into monohybrid is equal to dihybrid. Totally we get the 16 plants. 16 plants we get in dihybrid as well. 4 plants we get in monohybrid. Here 2 monohybrids are there. Automatically we get the 16 plants, in which how many plants are yellow, how many plants are round. Automatically we know yellow plants are 12 as well. Round plants are 12 because in monohybrid they are 3. In dihybrid they are 12. In monohybrid the green and wrinkles are single. Automatically in dihybrid they become 4 plants. 4 plants are there. So right. So here 9:3:1 is the ratio in F2 generation. 9 plants, 9 plus 1. These are the parental phenotypes. Parental types, right? Parental phenotypes. So 9 plus 1. 9 plants as well, 1 plant. Okay. So, okay, 9 plus 1, these are the 10 plants are parental phenotypes. Parental phenotypes nothing but round yellow as well wrinkled green. These are the round yellow. 9 plants are there. 9 is round yellow plants as well. 1 is wrinkled green plants. So these are the parental generations. Remaining these 3 plus 3, 6 plants are recombinants. Nothing but round green, which are not present in parental generation. And another 3 is wrinkled yellow. The round green and wrinkled yellow are not present in the parents but appeared in F2 generation. So they are recombinants. Recombinants, reunion to form recombinants, right? Round yellow 9, it is one of the parental generation. Wrinkled green also parental generation. And round green is the recombinant. As well, wrinkled yellow is the recombinant. These 6 plants are recombinants. Totally 6 plants are recombinants and 10 plants are parental combinants. 6 plants are recombinants and 10 plants are parents. So, right. It is the phenotypic ratio. If you go to genotypic ratio, right? How do we get the genotypic ratio? According to monohybrid class, 1:2:1 is the genotypic ratio of one monohybrid class. Genotypic ratio is 1:2:1. We take another monohybrid class also 1:2:1. These are the genotypic ratios of different monohybrids. They combine to form dihybrid. Automatically we get the 1:1, right? 1:1. Automatically 2:4:2 as well 1:1. So this is the genotypic ratio of dihybrid. Genotypic ratio of dihybrid 1:1, 2:4:2, right? 1:1, 2:4:2, 1:1, right? Totally, how many genotypes are there? Totally 9 genotypes are there. 9 genotypes. How many phenotypes are there? 9:3:3:1. So totally 4 phenotypes are there. 4 phenotypes are there, in which 9 plus 1 are parents, 3 plus 3 are recombinants, and 10 parental combinants as well, 6 recombinants, right? 4 types of phenotypes, but here 9 types of genotypes are there. 1:2:1, 2:4:2, 1:1. Totally 9 genotypes are there, which are formed by the different monohybrid genotypes. 1:2:1 is the round wrinkled monohybrid class F2 ratio as well. Another one is yellow green. F2 ratio is 1:2:1. Automatically we take the 2 characters in dihybrid at the same time. The result is 1:2, 1:2:1, 2:4:2, and 1:1 is the genotypic ratio. There are 9 genotypes are there. 9 genotypes are there, right? So for example, capital R, capital R, capital Y, capital Y, capital R, capital R, capital Y, small y, capital R, capital R, small y, small y, capital R, small r, capital Y, capital Y, capital R, small r, capital Y, small y, capital R, small y, small y, small r, capital Y, small r, capital Y, small y, small r, small y. So this chart helps us very easy to identify the how many parental combinations, how many recombinants, how many double homozygous, how many double heterozygous. So right, these are the 9 genotypes. Totally 9 genotypes are there. This is one of the genotype. So how many plants are there? This type of genotypes we know. Capital R, capital Y. So it is the round yellow plant. Round yellow plants. How many plants are there? Capital R. For example, capital R, small r, small r. It is the F2 ratio of monohybrid, in which homozygous round, how many plants are there? 1/4. Heterozygous round, how many plants are there? 2/4. Homozygous wrinkled, how many plants are there? 1/4. So automatically it is the dihybrid double homozygous. Here capital R, how many plants are there? 1/4 into 1/4. Totally 1/16. So round yellow are totally one plant. The genotypic shows one plant is there in 16. Right. So in monohybrid, homozygous round is one plant. Heterozygous round is two plants. Homozygous wrinkled is one plant. So here double homozygous is there. 1/4 into 1/4, automatically one plant is there. Such genotype shows only single plant. Right. So here, so it is the one plant. Here two plants are there because it is homozygous, nothing but 1/4. It is heterozygous. How many plants are there? 2/4. 2/4 is equal to 2/16. So two plants are there. So it is the another genotype, right? Homozygous 1/4, homozygous 1/4. Automatically 1/16 plants are there. Same genotype, right? It is 1. If we go to this, it is heterozygous 2/4 to homozygous 1/4. Automatically we get the 2/16. So two plants are there. 2/4 because of heterozygous as well. It is also heterozygous 2/4. Then automatically we get the 4/16. So these plants are present in dihybrid 4 2 generation. Right, 2/4 into 1/4, automatically 2/16. So these plants are 2. So 1/4 into 1/4 because of double homozygous, automatically 1/16. This plant is 1. 1/4 into 2/4, automatically 2/16. These plants are 2. 1/4 into 1/4, automatically 1/16. These plants are 1. Right. These are 2. These are 4. These are also 2. This is 1. This is 2. This is 1. 1:2:1. 2:4:2. 1:2:1. So these are the 9 genotypes totally. So 9 genotypes are there. So capital R, capital Y, one genotype. Capital R, capital Y, small y, 2 genotypes. Capital R, small y, 1 genotype. Capital R, small r, capital Y, small y, 4 genotypes. Capital R, small y, 1 genotype. Capital R, capital R, small y, 2 genotypes. Capital R, small r, capital Y, capital Y, small r, capital Y, 1 genotype. Small r, capital Y, 2 genotypes. Small r, small y, totally 1 genotype. So totally we get the nine genotypes. So 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 genotypes are there. So round yellow. So these 4, totally 9. 4, 1 plus 5, 2, 7. So one is missing, right? One, two, 4. Right, capital R, capital Y, capital Y, small r, capital R, small y. So it is so right. So capital R, small r, capital Y, capital R, small r, capital Y, small y. So I think so these 3. So this is the one. This 9 belongs to round yellow as well. Round green, wrinkled yellow, and wrinkled green. So right, 4 plants, right? This is the 2 plants, right? 2 plants, 4, 2. So totally 9, right? This is exactly right. These are the genotypes. So 1:2:2:4, 1:2:1, 2:1, right? Totally 9 genotypes are there. 9 round yellow, as well these 3 round green. These are the recombinants. As well these 3 wrinkled green. These are also recombinants. And wrinkled green, wrinkled yellow is recombinant, right? Wrinkled green is the parental combinants. So this is the parental combination. As well, this is also parental combination. Totally 9 plus 1, 10 parents are there. These 2 are 6 recombinants are there. This is the recombinants. These 9 plus 1 are parents. 9 plus 1 are parents as well. 6 are recombinants. So this is the dihybrid cross. We must know the genotypes. How do we get the genotypes? Right, totally. By two monohybrid crosses, automatically we get the dihybrid. Two monohybrid phenotypes, automatically we get the dihybrid phenotype. Two monohybrid genotypes, automatically we get the dihybrid genotype. There is no difficult. So it is very easy to understand. Very, very important also. Right. How many double homozygous plants are there? Double homozygous, capital R, small r, capital Y, small y. It is the double homozygous. How many plants are there? Nothing but 2 by 4 into 2 by 4. Automatically we get the 4 by 16. Double homozygous are there. Double heterozygous are there. How many double homozygous? Right. Capital R, capital R, capital Y, capital Y. It is the one plant. Capital R, small y is also one plant. Small r, capital Y is also one plant. Small r, small y is also one plant. These four are double homozygous plants. Like this, we can get any type of ratio. Right? These are the double homozygous ratios. Double homozygous. This is the double heterozygous. Double heterozygous are 4 plants. Double homozygous are 4 plants. So right. Then we get this ratio. We can calculate easily because of double homozygous. Automatically that is the one. Single homozygous, single heterozygous. Automatically 2 plants are there. Single homozygous, double homozygous. Automatically we get 1. Single homozygous, single heterozygous 2. Double homozygous 1. Double heterozygous 4. Single heterozygous, single homozygous 2. Single homozygous, single heterozygous 2. Double homozygous 1. We can get any type of ratio. So right. Next we go to independent assortment. According to based on the dihybrid cross, Mendel proposed independent assortment. So what is independent assortment? When two pairs of traits combined in a hybrid, segregation of one pair is independent of other pair of characters. That is independent assortment. Based on monohybrid cross, Mendel proposed law of segregation. Based on monohybrid cross, law of segregation is there. Law of segregation. Mendel proposed law of segregation. Based on dihybrid cross, law of independent assortment. Independent assortment. As well, based on monohybrid, other law is there, that is law of dominance. Law of dominance also. Right. Law of segregation says when two alleles in a hybrid, when two alleles present in a hybrid condition, capital R, small r, two alleles present in a hybrid condition, they do not show any blending at the time of gamete formation. They segregate independently so as to each gamete will get single allele. So automatically gametes are always pure because each gamete carry only one allele for one gene. Always gametes are pure because the gamete always carry haploid condition. Gamete always carry one allele for one gene. So it is the law of segregation. One allele enters into one gamete, another allele enters into another gamete. So according to monohybrid cross, Mendel proposed law of segregation. Right. Now dihybrid cross. So dihybrid cross, Mendel proposed law of independent assortment. Here in F1 generation, we get the capital R small r, capital Y small y. So this is the dihybrid. Dihybrid, we get the F1 generation, dihybrid. So right, here 4 alleles are there. 2 for one color, 2 for color, 2 for shape. There are totally 4 alleles are there. Each allele has the 50% of choice. Each allele has the 50% of the choice to enter into the gamete. Every allele has the chance 50% to enter into the gamete. Automatically, capital R enters into one gamete. Automatically, maybe capital Y, independent assortment. So, for same character, only one allele enters into a gamete. For one character, only one allele. Here, the character is round. Capital R, small r is there. But only one allele enter into one gamete. Already capital R is there. The capital R may go with capital Y. It is one of the combination. Capital R may go with small y. It is another combination. As well, small r may go with capital Y. Small r may go with small y. So this is independent assortment. Every allele has a chance to go with capital Y or small y. Capital R has the chance may go with capital Y, may go with small y. Independently assorted. Small r may go with capital Y, small r may go with small y. It is called independent assortment. When 2 pairs of traits are combined in a hybrid, 2 pairs of traits are combined in a hybrid, segregation of one pair of character is independent of the other pair of character. That is called law of independent assortment. Right. So at the time of meiosis, at the time of metaphase, so initially there are 4 chromosomes are there. So this is one chromosome. So this is another chromosome is there. So for example, so spindle fibers is formed and attached to the chromosomes. Automatically they push. So each cell gets the one chromosome as well. Another, for example, we gave the names. It is, for example, capital R. So it is the small r. It is the capital Y. It is the small y. For example, it is one of the combination. So capital R go with the capital Y. Capital R go with the capital Y. It is one of the combination. Another combination is so small y, small r. This is one of the combination we get. Right. Next, so right. One chromosome, one chromosome. Right. Here spindle fibers attached. Right. Spindle fibers are formed attached to the chromosomes. Right, right, right. So previously what we seen, capital R, capital Y enter into one gamete. It is one character, it is another character. Capital R go with capital Y, small r may go with the small y into different gametes. Capital R, capital Y, but so right. So for example, it is the small y. So capital R, capital Y, right. So it is the small r, it is the capital Y, capital R, small r. So at the metaphase, at the time of metaphase, the combination of alleles is varied. Combination of alleles is independent assortment. So initially it is another combination. We get the two gametes. Automatically one gamete get small r, capital Y, right. Another gamete small r, so capital R, small y. The capital R, small r, capital Y enters into one gamete, and capital R, small y enters into another gamete. It is one of the combination because at the time of meiosis, at the time of metaphase, the spindle fibers attached to the 50% of the chromosomes of the cell. Another pole, the spindle fibers attached to the 50% of the chromosome, automatically they separate the total two genomes. In this combination, for example, one chromosome, two chromosome, third. These are the haploid set of chromosome. This is one haploid set of chromosome. This is one haploid set of chromosome. So at the time of metaphase, spindle fibers formation, so right here, suppose for example, spindle fibers are formed from two opposite poles. One spindle attached to one chromosome, automatically the second spindle attached to the another chromosome. For example, if this spindle attached to the second chromosome here, automatically it attached to the here. For example, this attached to this, automatically this attached to third. Like this, totally 1 to 23. 23 chromosomes, 1 to 23. These are the two haploid set of chromosomes which came from father and mother. Every individual, every human being has two sets of chromosomes. At the time of meiosis, by formation of spindle, the two sets of chromosomes are separated by attaching the spindles. So like this. So here first chromosome enters to one side, automatically the another first chromosome enters into another gamete. Here the combination is changed. The second chromosome, for example, it is the father, it is the mother set of chromosomes. The second chromosome of father enters to one pole, automatically the opposite pole gets the another second chromosome. Like this way, totally 23 chromosomes comes to one pair and enters into one gamete. Remaining 23 chromosomes, nothing but haploid set of chromosomes, enters into another gamete. So this is the independent assortment. Like this, capital R may go with capital Y. Capital R may go with capital Y. Here, small r may go with capital Y. Capital R may go with small y. It is one condition. Capital R may go with capital Y. Capital R may go with small y. Small r may go with capital Y. Small r may go with small y. This type of condition. How four types of gametes are occurred? So at the time of gamete formation, haploid set of chromosomes enters into different gametes. It is because of by formation of spindles. Spindles attached to the one of the set of chromosomes. The attachment is independent. This attachment is independently assorted. That is why we get the four type of gametes: capital Y, capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Right. This is the independent assortment. It is very, very important concept. Independent assortment, right? So, based on the dihybrid cross, Mendel proposed the independent assortment. Independent assortments, when two pairs of traits are combined in a hybrid. When two pairs of traits are combined in a hybrid, segregation of one pair is independent of the other pair. So here it is the dihybrid. How many types of gametes we get? So capital R, every gamete, every allele has the chance to may go with the opposite allele, right? Capital R may go with. For example, capital R, small r related to the shape of the character, and capital Y, small y as the color of the color character, right? Color of the seed. Automatically one allele, every gamete gets two alleles, one for shape, one for color. So automatically capital R independently go with capital Y, capital R independently go with the small y, small r independently go with capital Y, small r independently go with the small y. So every allele has a chance. 50% of the choice may go with the other allele. This is called independent assortment. Independent assortment. It is proposed by Mendel based on the dihybrid cross. According to monohybrid cross, Mendel proposed segregation. Already we know what is segregation. So here also hybrid is there, monohybrid. Every allele get only may capital R or may small r. Every allele get only, every gamete get only one allele. So these are the gametes. Every gamete get only one allele, right, for one character. Here two characters are there. Every allele get two, every gamete get two alleles. Here two characters are there because that is dihybrid. These are the gametes. Every gamete get two characters for two alleles for two characters. So automatically every allele has a chance, independent chance to go with the other allele. Capital R go with capital Y, capital R go with small y, small r may go with capital Y, small r may go with small y. It is totally the independent assortment. It is very, very important concept. You must learn. Okay, dear students, if you got any doubt about this concept, please arise your question.
కొండమల్లపల్లి హ్యాండ్ రైజ్ చేశారు. మీ క్వశ్చన్ ఏందో అడగండి. అన్మ్యూట్ చేసుకోండి. క్వశ్చన్స్ ఎవరికున్నా కానీ దయచేసి అన్మ్యూట్ చేసుకోండి. ఆప్షన్ మీకు ఇవ్వబడింది. సో ఇప్పటి వరకు జరిగిన టాపిక్ లో ఎవరికైనా ఏమైనా క్వశ్చన్స్ ఉంటే దయచేసి క్వశ్చన్స్ అడిగితే సారు మీ డౌట్స్ క్లారిఫై చేస్తారు. స్టూడెంట్స్ whoever have the questions regarding the topic which have been taught, please unmute yourself. We have been enabled the option of unmuting yourself. Please, if you have any questions, ask to the sir. Kodamalepalli has raised the hand. Please unmute yourself and ask the question. लेबर का है. కొండమల్లపల్లి గర్ల్స్ ఉన్నారా? ఎవరికైనా క్వశ్చన్స్ ఉన్నాయండి? కొండమల్లపల్లి, హుస్నాబాద్, మిర్యాలగూడ, భద్రాచలం, కాటారం, వనపర్తి స్కూల్స్ ఎవరికైనా ఏమైనా డౌట్స్? ఓకే, టాపిక్ అర్థమైందా అండి? ఎవరైనా టాపిక్ అర్థమైందా సార్ చెప్పింది? ఇటు టాపిక్ అర్థమైందని చెప్పట్లేదు, అటు టాపిక్ డౌట్ ఉన్నాయని చెప్పట్లేదు.